Yeah, thank you for the introduction, and thank you for inviting me. I'm happy here to present you some results, and it's not easy now to switch from the atmospheres, and especially from the M-type stars, to this topic. I try to mention that there are these methanogene organisms which are using CO2 and H2, as mentioned just before by Michael. And we try to find out how to detect methane life forms, especially in a Mars-like environment. First of all, the source of methane is unknown. We have, Barbara has mentioned this in the last session, and there are a lot of publications going on, and there's a high debate about where is this methane coming from. We are not dealing now with the detection of methane, but on detection of life forms, methane producing life forms, and especially to see if we can add some information what we know on Earth studies, and especially in the field studies and in the lab, and perhaps we have then an approximation to detect these life forms. So the latest theoretical models appear to be a favoring source due to, not favoring source due to life activity, especially the mentioned publication of Sahne, what depends on the amount of methane. So we are really forced to detect life forms and to see if life is on the surface. And Raman methodology is very important. So NASA has developed the Mars Science Laboratory. I mean, it's changed now, the name. So a further MSL mission, and ESA, NASA, ExoMars, Trace Gas Orbiter will check then the amount of methane, the real amount of methane on the surface of Mars. And we know that the Raman methodology is also used. Francis is also involved in. So we just think we have to make a very systematical approach to see if these life forms are able to be detected. And this is our approach. We are using these following organisms. We have the organism just on the top, and I will focus during my talk on this organism. It's a new classified and new mentioned species. It's Methanosarcina gelisolum, and it's living in biofilms, as you can see here by the light microscope, and here also by the electron microscope image. It is from the Siberian permafrost, and it is also a halophilic organism. So what is also most important, because we have to think about that if water is also nowadays liquid on Mars, this depends on cryobrines and very salty environments. The sample preparation was done systematically, as I mentioned before, and we used glass slides first to distribute the organisms, and we must know that these glass slides have also a kind of Raman spectra. Then the organisms are embedded in salty environment like natrium chloride or natrium hydrogen carbonate, and these salt mixtures has to be also mixed with Mars analog substrates. So each of these steps were analyzed, and we have checked the Raman spectra, even of the methanogene themselves, so isolated and just put it on the glass slide, as well as in the mixtures of the halophilic environment and the Mars analog substrates. So we use a kind of sulfatic Mars regolith substrates and even also phyllosilicate Mars substrate. The experiment setup you can see here. We use a confocal Raman spectroscopy from the company VTEC Alpha 300 System 6. The objective is about 10 to use a 10 multiply so that you have the impression in the microscope. The excitation of 532 nanometers, and the power is about 1 milliwatt on a sample. So the energy, like it is foreseen for the ExoMars missions, and and the spot size is lower than 1.5 micrometers, but we have to take into account that the cells are about 1 micrometers, and so the resolution is not as good, and I will give you later on some information on that, why this can disturb some results in the interpretation of these results. So we are firstly checking these analyses. We are doing these analyses under room temperatures and at air ambient temperatures, so that lab conditions. But we are now at the stage that we are also doing this under Martian conditions, and this will be an outlook at the end of my presentation. Just to give you an overview how the spectra look like if you are using just the salt solutions, the dried salt solutions. So we have just natrium chloride and natrium hydrogen carbonate. Sorry, there's a mistake that's in the diagrams. And you can see that especially natrium hydrogen carbonate has very different spectra ranges, and natrium chloride is not as much very good to see, and we have to focus into that we can distinguish some spectra peaks. But this is a very good approximation if we are adding the methanogenes, because otherwise these spectra will disturb. And it seems if life is present, you have more fluorescent spectra, so fluorescence is disturbing Raman spectra. And so if life is present, this is dominating the spectra of all kind of salt concentrations. We have here different spots in the slide. You can see this is just glass, and here are the other salt concentrations, and you have some organisms still there, about 1 micrometer, as I mentioned, and also some biofilms. And the red curve is just indicating the spectra of the methanogenes, and you have here very distinctive spectra, and you have also sometimes appearing the amorphous carbon, as mentioned before, in the fluorescence range. Oops. And if we analyze just the methanogenes themselves, so on a glass objective holder, you can distinguish very easily the spectra of the glass light and the methanogenes themselves. And if you look here, we have a double peak at the position we just mentioned before at 2913 to 2913. The same is obvious if we use the mixture of mass simulation analog substrate, and we can then distinguish the peaks belonging to the methanogenes, the natrium chloride, even some elements which are in the mixture of the mass, sulfatic mass environment, phosphoride, and then this peak where you can a little bit see the double peak in this range. What is important to know, we have heard a lot about amorphous carbon or not really good detectable carbon contents, and this is every time appearing even if we are analyzing the methanogene organisms, and you have it here. So we mapped it and we analyzed where are the methanogenes, and we saw also some spectra of the hematite of the S-mass analogon. Line scan just showed us that the amorphous carbon is appearing and disappearing, and according the line we are going. We are thinking about, at the first, when we have discovered this, we have thought about if this could be due to the membrane and there's a periodicity. If we're going with higher lines in between in the samples, there's a periodicity. And we thought maybe that when the membranes are coming together, you have a higher carbon content, and therefore, but this can't not really be true because also of the resolution. We have then to need a new resolution, a better resolution, but it's really interesting to know that the carbon is mostly connected, but we are not sure if we can really, we are sure if this appears together with the other spectra, which appears every time, then it belongs to life forms and especially on methane-producing organisms. So I will finish with these conclusions that so it is, the Raman might be a suitable method to detect terrestrial methane-producing archaea. I emphasize this is terrestrial archaea, but maybe that on Mars there's an interconnection to Earth life, so that we are able to detect this with Raman. And the question still remains, is this valid really for Mars inhabiting microorganisms and Mars environment? But it is an approximation, and our Earth life is still the reference system we use. So the methanogens are also able to be detected in salt concentrations and in Mars analog mixtures. Another thing what we are doing now, and this is the outlook before I'm ending, we have a Mars simulation chamber providing the gas compositions, a mixture of gas compositions. There's a gas mixing system with 95% CO2 and the rest in nitrogen and argon. And we are cooling this system. We can cool it down to 4 Kelvin, so largely down and not only to Martian conditions, but we have also the possibility, as you can see, to do this under Martian conditions. And we are using then a vacuum pump so that we have a level about 8 millibars and sometimes reach also 6 millibars. And you can really see that, especially if we are using these salts, that the spectra changes according to the temperatures. And this means, and also the environmental conditions, this means that we have to work also on this if we want to detect something under Martian conditions, because the spectrum might change. And with this, I think.